First of all, well, thank you for being here. Thank you for the foundation for selecting this work, and thanks also for the families, to the families for funding this research. So, well, I will be talking today about our attempts to increase the throughput of the PMCA technology, this prion amplification technique, with the aim to look for compounds able to inhibit prion replication, but we want to take a focus on specific prion strains. And I will tell you, I hope to convince you why this is really important to consider. Well, we have been hearing today, I think that it's all clear for us about these two proteins. These are cartoons of what the structures, that just simplistic representations of how these proteins that are important for prion diseases are present. So we have, on the one hand, the PRPC, the normal prion protein that all of us generate, and on the other hand, the PRPSC, that is the disease-associated form, that under certain circumstances, this normal protein misfold, change its conformations, its shape, and then it induces disease. So again, in a very simplistic way to explain this process, if we take this protein and we put it in the test tube, in water, eventually this protein will have will aggregate. It has the tendency to aggregate. Now, we know that if this process is not easy to occur sometimes, it takes some time. But if you add a preformed aggregate or seed, that we called in the field, these events will occur much faster. And actually, this explains very well why prion diseases are infectious, basically. If you introduce a seed into your body, you will catalyze this reaction, and this protein will start to aggregate at expenses of the normal protein that all of us generate. The problem is that the infectious prion protein, the disease-associated prion protein, it doesn't misfold it doesn't change its conformation in a single way. It can take different shapes. This is the basis of what we call prion strains. So in definition, the main difference between prion strains lies in the conformation that the disease-associated prion protein acquire. So these differences in the shape of the disease-associated prion proteins are easy to reconcile if we consider different animal species where there are differences in the amino acidic sequence. So the building block of the protein, will be different. So the shape, all these building blocks will fit differently into the final shape. Now, interesting thing on prions is that within a single conformation, a lot of different shapes can be generated, and each of these shapes can lead to a different disease. So I want to focus on this part of this slide that explain what are the disease consequences of these different strains. For example, if we have these hypothetical two strains A and B, they can induce the disease in a faster or a slower manner. They can develop different clinical signs. For example, in experimental animals, in hamsters, for example, we can have clinical signs that varies from hyperactivity to drowsiness to hyperphagia, so the animals, in some cases, the animal cannot stop moving, and the other one, the animal cannot stop eating, so the clinical signs are quite different. And this is related to different properties of these different prion strains. One of them is what we call prion tropism. Different strains have preference to go to target specific regions of the brain. And different prion strains also have different degree of toxicity. They are able to kill neurons or damage synapses faster compared to others. So in that sense, focusing on prion strains is quite relevant in different aspects: how we understand the disease, how we diagnose the disease, and how we can cure them. Okay? So previous evidence suggesting that the difference between prion strains and conformation were linked was indirect, So we based a lot in biochemical analysis, which saying that, okay, by this biochemical property, these two different strains are different, so the structure of the protein could be different. Fortunately, and in recent years, by studies by the group of Dr. Byron Cowie and others, we know we have a more certainty about the structure of the infectious prion protein. And something that we also know is that now the structure of the different strains is different. So what are the implications for this in drug discovery? So let's take the druggable site, a site of the protein that we can modify for therapeutic purposes. If we take the strain A and strain 2, strain 1 and strain 2 that have different conformations, and we take a drug, that is working great for strain 1, we'll say, okay, so this this compound may look promising, for a treatment for prion disease. But then if we test the same compound in another strain that have a different conformation, perhaps this drug will not fit, And in that sense, the drug that is working great, for example, in mouse, it will not work so great for humans, And this is a big problem. Or if we have a drug that is working very good for one specific human prion strain, it may not be working properly for a second human prion strain. So for that reason, it's really important to consider prion strains when we are looking for potential treatments, if we are focusing on the misfolding of the prion protein. So we have different approaches to look for potential treatments. And we have different targets, as was previously mentioned by Dr. Knight. So one thing that is, I think, a consensus is that the central event in prion diseases is the conversion of the misfolding of the prion protein, this conformational change of the normal prion protein into the abnormal isoform. And to study that and to potentially test potential drug candidates, we have different avenues. I will say that the optimal, is to go for, of course, in the case of humans therapies, is to go to clinical trial. The issue is that how do you select for a specific candidate to go for that trial? So one way to go, it will be to do bioassay, to test animals, animal models that either express the human prion protein or prion protein for other animal species. The problem is that these experiments are very long and they are very expensive. Basically, they are not practical to screen multiple candidates, Another one that you can say, okay, is easy to go are cell cultures, And there are several systems that are able to replicate prions based in cell cultures. Again, as Professor Knight mentioned before, this is more simplistic, but still is a fair screening tool. The problem is that cell cultures do not respect very well this strain issue. Several cell cultures that are able to replicate mouse prions, they are not able to replicate all the spectrum of mouse prions, and the same for other animal species. So in that sense, it's not optimal. Now, we go to the amplification assays. The amplification assays are faster, are comparably cheaper compared to bioassays, and they can replicate a wide array of prion strains. We have two assays, the PMCA and RT-Quick. In the laboratory, we are working on PMCA. I will explain the reason why. Now we have a certain preference for this assay, for this particular experiment. So I will develop a little bit more on PMCA. So PMCA is a technique that basically what it does is to mix, to have in a test tube, a sample that it contains little quantities or is expected to contain some aggregates, again, or seeds that we call in the field. And these are combined with an excess of the normal prion protein. So when we incubate that, these fibers, these seeds recruit normally folded proteins into the growing aggregate, but these aggregates are only active at the end. So in order to increase the efficiency of the conversion, we apply some force, to break these fibers and to create new active ends that will recruit more normal protein into the abnormal fiber. And this, if we repeat this process of incubation and sonication in a cyclic way, we will have a partial depletion of the normal prion protein into the misfolded aggregates. So RT-Quick is another technique that operates in a similar principle. The problem with RT-Quick is that it doesn't generate infectious material. This is good in terms of screening, so if you are screening a lot of samples, you don't want to amplify the infectious material in your laboratory. However, for this, the problem if RT-Quick is not generating infectious particles is that it's not respecting the strain properties, and we know that PMCA is able to do that. So this is the conventional assay that we have for PMCA. This is the platform that it takes. We do it in PCR tubes. We use 100 microliters of reaction to do the assays. These are other features. It takes around 6 days, and the total time per round. And it takes 6 days in total for the 3 passages of PMCA to increase sensitivity, as Dr. Barria showed before. And the total time is around 8 days. So this PMCA is able to be used to the screening of multiple samples. However, it's not practical, again, to screen drugs, a large library of compounds. So the problem that we had here is that prion diseases are caused by different prion strains, and this, as I told you, may be a problem when identifying anti-prion drugs. And our goal is to develop an in vitro screening system to identify strain-specific anti-prion molecules. For that, what we are trying is to miniaturize, to make a PMCA more suitable for high-throughput screening by standardizing it in the different platforms, and also, once this is achieved, the idea is to test libraries and try to identify strain-specific anti-prion compounds. So I summarized a lot of work that we have done in this single table, and please believe me that it doesn't make justice because it was a lot of work. So, for example, when we try to go from PCR tubes to 96 well plates, not all the plates, it's not just to take a plate and to do it. The different plates have different materials, the efficiency is different, so it was a lot of work. We were able to identify the specific plates. We were able to reduce the volume to have reproducible results. We were able to reduce a lot of the components of the PMCA, but most importantly, we were able to reduce the whole process to 2 days. So this is an example of how this 96 well plate format is working at this moment, and how it's working for different prion strains. In here, as a proof of principle, we did it in mice and Syrian hamster. We took 3 different strains of each animal species coming from different origins, etc., that behave completely different in vivo. And as you can see here in this cartoon, the black dots represent seeded reaction, so they should be positive. The white ones, unseeded reaction, they should be negative. And you can see that the PMCA with the optimized system is performing very well. And actually, we optimized it to avoid false positive and false negative. That was a lot of work too. But this is the final result for these different prion strains. So after we achieved that, what we did was to test preliminarily a small library of compounds that are known to have, that were reported before as anti-prion, with anti-prion activity, or with anti-amyloid activity. Anti-amyloid meaning that they were affecting other misfolded proteins, amyloid beta, alpha-synuclein, etc. And as you can see here, this is how the screening looked like, but the results can be better appreciated in the table. We tested each compound at 3 different concentrations. As you can see here, some compounds were not able to inhibit these prions. Some of them were previously shown to inhibit prion replication. In our system, for these particular strains, they didn't. But as also you can see that some strains are able to deactivate only to certain prion strains, but not to others. So we are seeing compounds that are very specific for particular prion strains. We are also able to detect the specific activity for specific compounds. Some of them were more efficient at lower concentration, some others are higher concentration. We did the same with hamsters. And basically, we observed the same effect that we observed with humans. So I think that we are in a good shape to screen compounds, in a strain-specific manner. And we are getting good results with that. So considering that, now we jump to a larger study. So we are testing now mid-size compound library, well, still small, 1,600 compounds, tested at a single concentration, 100 micromolar. We want to do that for different human prion strains, for different deer prion strain. Why deer? Because we want, this is a very relevant disease. It's the most relevant animal prion disease in North America, and also as a proof of principle to test whether for animals and humans there are also differences in the compounds that are active. So at present, over 800 compounds have been tested for CWD, for a single strain of CWD, and 24 hits have been identified. We expect to reduce that as we are decreasing the concentration of compounds on the hits. And the current progress, we successfully modified the PMCA technology to increase the throughput. That was validated in 6 experimental prion strains. The PMCA system confirmed the prion strain-specific effect of several previously described anti-prion and anti-amyloid molecules. And as I showed you, we initiated the screening in the, to identify molecules active against a prion strain affecting deer. The future plans for the next year are to conclude the screening of deer prion strains, conduct secondary analysis on hit compounds to increase chances of success in preclinical tests, of course, evaluate this in human prion strains, and efforts are being made now to set up this technique in 384 plates. This has been very difficult to increase the throughput of the technique. So I will jump to that. I want to like to thank the people that is working on this, specifically to Rebecca, who is doing most of the work in this front, and to the families that provided funding for this work. And thank you all for your attention.